Schizophrenia is a chronic and severe mental disorder characterized by profound disruptions in thought processes, perceptions, and behavior. Schizophrenia can severely impair quality of life, and current treatment methods such as antipsychotics have significant limitations. Research shows that up to 30% of patients don't respond to antipsychotic treatments, while others experience residual symptoms or adverse effects that impact their daily lives. This has led researchers to explore alternative therapies, and one emerging option is transcranial direct current stimulation, or TDCS. TDCS is a non invasive, affordable, and portable treatment method which uses weak electrical currents to stimulate specific brain regions. It shows promise in reducing negative symptoms and auditory hallucinations, which can be resistant to traditional treatments. Several studies have examined the effects of TDCS in patients with schizophrenia. For example, Alman and colleagues in 2018 found that TDCS improved negative symptoms such as a lack of motivation and emotional flatness. Brunelli and colleagues in 2012 also reported significant reductions in auditory hallucinations. In their study, participants experienced a 31% reduction in hallucinations, and this effect lasted up to 3 months. Kim and colleagues in 2019 showed that improvements in symptoms were linked to the number of TDS sessions, with more than 10 sessions leading to better outcomes. However, studies like those by Yang and colleagues in 2019 indicate mixed results for positive symptoms, which included hallucinations and delusions. Despite these promising results, there is significant variability in research findings due to the differences in how studies were conducted. Some studies use different session lengths, electrode placement, and patient profiles, making it difficult to compare direct outcomes. There's a growing consensus that personalizing TDCS treatments based on individual factors is crucial for its success. To understand how TDCS might help treat schizophrenia, it's important to look at the underlying physiological mechanisms of the disorder. Genetics play a big role, accounting for about 80% of a person's risk of developing schizophrenia. Some key genes involved include COMT, DISC1, and NGR1, which both affect brain development and how neurotransmitters are regulated. These are shown specifically via the table on the screen. There are also environmental factors, things like inflammatory processes during fetal development, immune activation, and birth complications, such as hypoxia or preterm birth, which can disrupt brain development. This leads to changes in neural migration and causes excessive brain pruning during adolescence, affecting synaptic density. Schizophrenia symptoms are driven by neurotransmitter imbalances. For example, too much dopamine activity in the mesolimbic pathways lead to positive symptoms like hallucinations, while too little dopamine in the prefrontal cortex contributes to negative and cognitive symptoms. This dopamine imbalance happens because of many complications with dopamine production and release, particularly in the striatum. Another factor is the NMDA receptor, which is a type of glutamate receptor. These receptors don't function properly in individuals with schizophrenia, leading to problems with cognitive and negative symptoms. Dysfunctions of these receptor in GABAergic interneurons cause too much glutamate activity, leading to an upset in the balance between excitatory and inhibitory signals in key areas like the prefrontal cortex and hippocampus. Reduced GABA activity in the prefrontal cortex is linked to cognitive deficits common in individuals with schizophrenia. Patients with schizophrenia also show brain abnormalities such as reduced grey matter in the prefrontal cortex, hippocampus, and temporal lobes, along with enlarged ventricles. This impacts their ability to think, plan, and remember. Additionally, neuroinflammation and oxidative stress damages neurons, which worsen cognitive dysfunction. These, combined with factors, explain why schizophrenia includes such a wide range of symptoms, from psychosis to cognitive issues to emotional flatness. So, how does TDCS target these physiological issues? TDCS works by using electrodes placed onto the scalp to send mild electrical currents to specific brain areas. This current changes the activity of neurons by altering their resting membrane potential. There are two types of stimulation: anodal and cathodal. Anodal stimulation makes neurons more likely to fire by depolarizing them, while cathodal stimulation does the opposite, reducing neuron activity by hyperpolarizing them. These effects are achieved by moving sodium and calcium ions in and out of cells, which activates ion channels. One important effect of TDCS is that it triggers changes in synaptic plasticity, specifically in how calcium activates pathways related to learning and memory. It also influences neurotransmitters, increasing glutamate activity and decreasing GABA inhibition, which can enhance neuron excitability. Additionally, it affects monoamines like dopamine and serotonin, which are crucial for mood and cognitive functions. At a molecular level, these changes activate transcription factors that regulate gene expression, leading to longer lasting changes in how neurons communicate and function. Now, why is TDCS useful in treating schizophrenia? Well, it helps to rebalance disrupted brain networks and neurotransmitter systems. For example, nodal stimulation of the dorsolateral prefrontal cortex can increase neuron activity in that region, which is often underactive in schizophrenia. This improves cognitive functions like decision making and working memory, helping to balance negative symptoms. On the other hand, cathodal stimulation of the temporoparietal junction can calm overactive brain areas linked to auditory hallucinations. By reducing this overactivity, TDCS can help restore the balance between excitatory and inhibitory signals, making hallucinations less frequent and less intense. In fact, the randomized controlled trials, as shown on the slide, have shown that cathodal stimulation of the TPG significantly reduces the severity of auditory hallucinations. These findings support the idea that TDCS can help reconnect brain networks disrupted by schizophrenia, leading to better symptom management. Moving forward, there are several areas of research to focus on. First, it's crucial to develop standardized treatment protocols so that findings can be compared across different studies. Second, as we've seen, personalization is key. Identifying biomarkers that can predict a patient's response to TDCS will allow us to tailor treatments more effectively. It's also important to explore the long term effects of TDCS and whether combining with other therapies such as CBT or medication could lead to better outcomes. And finally, more research is needed on how TDCS affects cognitive deficits in schizophrenia, which are often harder to treat than positive and negative symptoms. In conclusion, TDCS has emerged as a promising alternative treatment for schizophrenia, particularly for reducing negative symptoms and auditory hallucinations. While there are still challenges in terms of standardization and personalization, the potential benefits of TDCS are substantial. Ongoing research will help us refine the treatment and unlock its full potential, providing new hope for patients who struggle with limitations of traditional therapies.